We're going to take a look at the distal convoluted tubule and collecting duct in this lecture. So the filtrate that enters the distal convoluted tubule from the loop of Henle, we've absorbed 80% of the water and 90% of the solutes, but we still need to reabsorb up to 19% of the water. So we're going to do our final urine concentration here in the distal convoluted tubule and collecting duct. The osmolarity of the filtrate that's coming into the distal convoluted tubule is at 100 milliosmolar, so it's not very concentrated. So it is a smaller volume than before we went through the loop of Henle, but it's much less concentrated. So if you were to urinate that out, it would be very clear and really no yellow color to it. The distal convoluted tubule and collecting duct are really different from the proximal convoluted tubule and loop of Henle because their permeability to water and solutes is going to be regulated by hormones. ADH is going to regulate water permeability, and aldosterone and atrial natriuretic peptide, or ANP, are going to regulate solute permeability. So the distal convoluted tubule and collecting duct are not permeable to water or solutes unless hormones are there to change that. And so the distal convoluted tubule and collecting duct, as we said before, it's going to be really important to reabsorb that final up to 19% of the water, and that'll give you the final urine concentration and volume. So that'll be determined here. In the epithelial cells that make up the walls of the distal convoluted tubule and collecting duct, there are tight junctions between the cells, but they are true tight junctions. So they're not leaky tight junctions like we saw in the proximal convoluted tubules, so you're not going to have things moving in between the cells. Let's take a look at water reabsorption in the distal convoluted tubule and collecting duct first. So how much water is reabsorbed will affect the blood volume and therefore the blood pressure. And it's also going to affect the blood osmolarity. And remember, the osmolarity is just talking about the solute concentration. So the distal convoluted tubule and collecting duct are impermeable to water unless ADH is present. And remember that ADH is secreted by the posterior pituitary gland. And ADH will be secreted in response to increased blood osmolarity, so that a lot of times is caused by dehydration, if the blood pressure is decreased for whatever reason, and also in response to another hormone called angiotensin II, which we're going to look at later. Remember that ADH stands for antidiuretic hormone. So that means ADH will tell the kidneys to save water from the urine and reabsorb that back into the blood. And that'll increase the blood volume and increase the blood pressure. So how ADH does this is it's going to tell the cells that are making up the walls of the distal convoluted tubule and collecting duct to put out aquaporins in their apical membrane, and that'll allow the reabsorption of water as the collecting duct goes down through the renal medulla through that medullary osmotic gradient. Remember that medullary osmotic gradient was really important. That was produced by the loop of Henle of the juxtamedullary nephrons. And so as water goes down and the osmolarity is increasing down through the renal medulla, water is going to be reabsorbed out of the filtrate and put back into the blood by osmosis. And so this will have you urinate a smaller volume of urine, and it'll be much more concentrated. It'll be much darker yellow in color. And then we'll take a look at what happens with diabetes insipidus, which remember that is either the person is not producing ADH or the kidneys are not responding to it. So here's our picture again of the nephron. And we're going to be taking a look down here as our collecting duct is going down through the renal medulla. And if ADH is released, it will tell these cells that are making up the walls of the collecting duct to put out these aquaporins on the apical membrane. And remember, the apical membrane faces towards the lumen or towards the urine that's being made. And once you put those aquaporins out there, now water can be reabsorbed. So we start with our filtrate having an osmolarity of 100 milliosmolar. And so as it comes down through the rena medulla, so let's say we're here at the 900 milliosmolar level. So water is just going to follow the solutes, right? So there's a lot more solutes out here in the rena medulla. So water is just going to go down its concentration gradient by osmosis, and then that water will be put back into the blood, and that'll increase the blood volume and increase the blood pressure. And as water moves out of the urine, that's going to create a smaller volume of urine, and it's going to be much more concentrated. The highest concentration you can get in the urine is up to 1400 milliosmolar because that's the highest osmolarity in the renal medulla. And this is just a picture just showing the ADH, which was released from the posterior pituitary gland. It's a protein hormone, so remember those are hydrophilic. They're going to bind to a cell surface receptor here on the collecting duct, and this happens to be a G protein linked receptor. We activate our enzyme here, make our second messenger. We end up phosphorylating some proteins which cause aquaporins to be put on that apical membrane, and now water can be moved through the epithelial cell and be reabsorbed back into the blood. And by reabsorbing that water, you're going to increase the blood volume and increase the blood pressure. And then also the urine volume will decrease and the urine will become more concentrated. So here's what happens when ADH has not been secreted. So remember that the distal convoluted tubule and collecting duct are not going to be permeable to water unless ADH is present. So if you have not secreted ADH, the urine will just flow out without reabsorbing any of the water. So it'll stay very dilute. So it'll be very clear in color, not really any yellow. And you'll have a large volume because remember this last step, you need to reabsorb about 19% of the water here. And if you don't do that, you're going to have a large volume that you're urinating. If you are secreting lots of ADH, maybe it's a hot day and you're dehydrated, then if you have lots of ADH being released, you're going to reabsorb a lot of the water from the urine. And the maximum concentration of your urine that you can make is 1400 milliosmolar. So that would be a really dark yellow, and you'd be urinating a very small amount of urine. And then in diabetes insipidus, this is what happens when you're either not secreting ADH or if the nephron is not responding to the ADH. So they will urinate large volumes, and then they have to drink a lot to keep up with all the urine that they're losing. This slide just shows the stimulation of ADH release. So maybe we are dehydrated and increase the osmolarity of the blood. So osmolarity is the solute concentration, and if you've lost a lot of water, the solutes become more concentrated and the hypothalamus will sense that and release the ADH to save more water from the urine. Or if the blood pressure is decreased, that will also be sensed and tell the posterior pituitary to release ADH. So remember that the distal convoluted tubule and collecting duct are not permeable to water or solutes unless hormones are around. So ADH we just talked about, that's going to control the permeability to water. And then permeability to solutes are going to be controlled by two other hormones, aldosterone and atrial natriuretic peptide, or ANP. Let's take a look at aldosterone first. Remember that aldosterone is secreted from the adrenal cortex. It's one of these steroid hormones, and it's going to be stimulated to be released by the renin-angiotensin-aldosterone system, or RAS. And RAS gets stimulated if the blood pressure is low or if there's low sodium in the filtrate in the urine. And then the other thing that stimulates aldosterone release from the adrenal cortex is if the potassium levels are too high in the blood. Aldosterone is going to stimulate the distal convoluted tubule and collecting ducts to reabsorb sodium from the filtrate and put that back in the blood. And in exchange, we're going to secrete the potassium ions. And if you're reabsorbing sodium ions, and if ADH is present, then water will just follow the solutes and will follow the sodium. And so you end up increasing blood volume and increasing blood pressure. So the effect of aldosterone is it will increase blood volume and blood pressure, and it also keeps the sodium and potassium levels in homeostasis. Here's what aldosterone does. So aldosterone will tell the epithelial cells making up the walls of the distal convoluted tubule and collecting duct to put more sodium-potassium pumps on their basolateral membrane, and then to put some sodium and potassium leak channels on the apical membrane. So if you're pumping sodium out of the cell, then sodium will leak into the cell down its concentration gradient, and then it just gets pumped out. So we're going to be reabsorbing sodium. And if ADH is present, which if your blood pressure and blood volume are too low and you're secreting aldosterone, you're also going to be secreting ADH. So if you're reabsorbing sodium ions, water is just going to follow those sodium ions. And so aldosterone will trigger an increase in blood volume and blood pressure through that way. And then the potassium is going to be pumped into the cell, so we'll have high concentration inside the cell and much lower in the urine, and so potassium will just diffuse down its concentration gradient and be secreted into the urine. So aldosterone is going to cause sodium to be reabsorbed, and then secondarily we're going to reabsorb more water because we have sodium being reabsorbed, and then we're going to secrete potassium. So the end result of aldosterone is it will increase blood pressure and blood volume, and it also will make sure the sodium and potassium levels are kept in the normal range. And this is just a picture of aldosterone. Remember, aldosterone is a steroid hormone. It's hydrophobic, so it just diffuses right into the target cell, binds with a receptor in the cytoplasm, and remember, it's going to turn some genes on and off, and so we're going to turn on genes and say, tell the cell to make more sodium potassium pumps and also to make some sodium and potassium leak channels. So we end up reabsorbing the sodium that gets put from the urine back into the blood, and then we're going to be secreting potassium, putting that out into the urine. Now we're going to take a look at the RAS system, and this all starts with renin. And renin is going to be released from the granular cells, which are part of the juxtaglomerular apparatus. And remember, these granular cells are wrapped around the afferent arteriole of the nephron. So what stimulates renin to be released is when the blood pressure drops. And when the blood pressure drops, the afferent arteriole that these granular cells are wrapped around are not stretched as much, and so that will trigger them to release the renin, which will increase the blood pressure and get that back to normal. We're also going to be secreting renin in response to sympathetic stimulation with our baroreceptor reflex when the blood pressure gets too low. And then also, if the blood pressure is too low, we're not going to be filtering out as much of the plasma at the glomerulus, so that'll decrease the GFR, or glomerular filtration rate. And so that means we're going to have less sodium and chloride ions in the filtrate, and so when that gets to the distal convoluted tubules, where the macula densa cells are, they will sense the decrease in the salt, and they will tell the granular cells to also release renin. Here's a picture of the RAS system. This is really important to know, so make sure that you can draw this out. I obviously will not make you draw this on the test, but I could ask, you know, if you're at this step, what happens next? And so make sure you know this picture. The other really big thing to take away from this is that the end result of stimulating the RAS system is that it increases blood volume and increases blood pressure. So that's why we're going to stimulate RAS, is if we need to increase blood pressure and blood volume. So we're going to start with the liver. The liver makes a protein called angiotensinogen that's released into the blood. It's inactive, it's just floating around, and it will be activated by renin. And renin is going to be released by the granular cells that are wrapped around the afferent arteriole in the nephron, and remember that they're part of the juxtaglomerular apparatus. And what stimulates the granular cells to release renin will be a drop in blood pressure, increase in sympathetic activity, or a decrease in the salt in the filtrate that's going to become the urine. So the macula densa cells sense that. So renin will be released, and it's going to convert angiotensinogen into angiotensin 1, and that'll happen in the blood. And then the other thing that happens with renin, just kind of a side note here, it will constrict the efferent arteriole, and if you do that, that will back blood up into the glomerulus, and then that will increase the blood pressure in the glomerulus, and that'll increase the glomerular filtration rate back up to normal because it's too low with the blood pressure being low. And then angiotensin 1 will be converted into the final active form, angiotensin 2, and this happens from an enzyme called ACE, which stands for angiotensin converting enzyme. This enzyme is located on the capillary walls of the lungs. So as the blood travels through the lungs, that ACE enzyme will convert angiotensin 1 into angiotensin 2. So angiotensin 2 has a lot of effects. It will go to the posterior pituitary and stimulate that to release ADH. Remember, ADH will tell the kidneys to save water from the urine and put that back into the blood, and that'll increase blood volume and increase blood pressure. Angiotensin 2 also causes vasoconstriction, so that will increase the blood pressure. And then angiotensin 2 will also go to the hypothalamus, where there's a thirst center, and it will make you thirsty. So if you drink more water gets absorbed from the GI tract, and that'll increase the blood volume and increase blood pressure. And then the fourth thing that angiotensin 2 does is it goes to the adrenal cortex and stimulates it to release aldosterone. Remember, aldosterone will go to the distal convoluted tubule and collecting duct and tell those cells to reabsorb sodium and secrete potassium. And if you're reabsorbing sodium from the urine and putting that into the blood, then water is going to follow that because ADH is also being released here as well. And so aldosterone will, as the end result, will increase blood volume and increase blood pressure. And then this is just a picture from your book, pretty much the same thing. And then this is just showing you the effects of angiotensin 2 to act to increase the blood pressure. So these are the four things it does. It's a vasoconstrictor. It tells the adrenal cortex to release aldosterone, tells the posterior pituitary to release ADH, and also tells your hypothalamus to make you drink more water. The other hormone that controls solute permeability of the distal convoluted tubule and the collecting ducts besides aldosterone is atrial natriuretic peptide, or ANP. So ANP pretty much does the opposite of aldosterone. So aldosterone, the end result is to increase blood volume and increase blood pressure. So it'll be released when the blood pressure is too low. ANP is going to be released when the blood pressure is too high. So it will end up decreasing the blood volume and decreasing the blood pressure. So as the name says, ANP is going to be secreted by the atrial cells in the heart. So they will sense when the blood volume and blood pressure are too high because the atria become too stretched, and that will tell them to release ANP. And ANP has several effects. So it will go over to the afferent arteriole and dilate that and then constrict the efferent arteriole. So by doing that, you're bringing more blood into the glomerulus but not letting as much out, and that'll increase the blood pressure in the glomerulus, and so you're going to filter out more of the plasma, and you end up filtering out obviously more water and more sodium. And then in the distal convoluted tubule and collecting duct, it will decrease sodium reabsorption. So it's going to close some of the sodium channels, and it will also inhibit the RAS system, so it'll inhibit renin and aldosterone secretion. And so that will decrease the amount of water that's reabsorbed. And that will help decrease blood volume and decrease blood pressure. So here's a picture of what AMP is going to be doing. So the first thing it does is it will dilate the afferent arteriole coming into the glomerulus, and then it's going to constrict the efferent arteriole that are exiting. So all of that will increase the blood pressure in the glomerulus. You're going to end up filtering out more water and more sodium. And then when you get over here to the collecting duct, AMP is going to do a couple things. So it will close the sodium channels. So we're not going to be reabsorbing sodium anymore. So we're going to leave the sodium in the urine. And that's where we got part of the name of atrial natriuretic peptide. Natriuretic just means you're peeing out sodium. So sodium doesn't get reabsorbed, it stays in the urine, you excrete that. And if you're not reabsorbing sodium, water's not going to follow. So you're not going to reabsorb as much water, you'll urinate more of the water out, and then that will decrease the blood volume and decrease the blood pressure. And then the other thing that AMP does is it inhibits the RAS system, because remember RAS wants to increase blood volume and blood pressure. And you're releasing AMP because blood pressure is too high and blood volume is too high. So AMP will also inhibit the release of renin and also aldosterone. And then these are just the effects of AMP, just showing you how it will dilate the afferent arteriole, constrict the efferent arteriole, that'll increase glomerular filtration rate. We end up filtering out more sodium and more water. And then it also is going to decrease the amount of sodium that's being reabsorbed by blocking those sodium leak channels in the distal convoluted tubule and collecting duct. And then also it's going to decrease the stimulation of the RAS system, so we're not going to be releasing renin and aldosterone. The last thing to talk about with the distal convoluted tubule and collecting duct is this is another place that we can regulate blood pH with the kidneys. So remember, we also did this in the proximal convoluted tubule. So we can secrete hydrogen ions. If we have too many, put those into the urine. And if we're too acidic as well, besides secreting hydrogen ions, we can also reabsorb bicarbonate ions and put those back in the blood. And then obviously if you're too basic, we just wouldn't secrete the hydrogen ions and we wouldn't reabsorb the bicarbonate ions. This is the picture we've been drawing, showing secreting the hydrogen ions into the urine. If you've got too many, you can pee those out. And then we can also reabsorb bicarbonate ions if we're too acidic as well, and that'll help increase the pH. So we can regulate the blood pH in the distal convoluted tubule and collecting ducts, and then the other place we did that was in the proximal convoluted tubule. And then this is just showing you acid-base balance. Remember that the kidneys are involved controlling hydrogen ions and bicarbonate ions. And then remember the respiratory system, the lungs are going to control the carbon dioxide levels. And this is just showing you where some of the hydrogen ions are coming from. We do get it from our diet. Proteins have amino acids, and then fats have fatty acids. Those are all acids that release hydrogen ions. And then also during metabolism, as we break down our food to make the ATP, we generate carbon dioxide, which with that equation we learned about in the respiratory system that can generate hydrogen ions, and then we can make lactic acid during fermentation, maybe we're sprinting or lifting weights. And then keto acids are made during aerobic respiration, and also as we're making amino acids they can be made as well. And this is just kind of a summary of what's happening in each part of the nephron, so you can review that. And then again, just what's happening in different places in the nephron. And then all I just wanted to show you with this table here is that if you take a look at the concentrations of these things in the urine versus that's in the blood, you can see valuable things like glucose. There's pretty much no glucose left in the urine. We're going to reabsorb about 100% of that. And then also other valuable nutrients, proteins, remember they're too big to fit through the gaps in the capillary walls that are in the glomerulus, so there shouldn't be any proteins in the urine. And then you can see our waste products here. They're going to be much more concentrated in the urine, and we've gotten rid of them from the plasma.